in my previous video, I have talked about the protein synthesis, how it is made in ribosome. I have discussed about this in details. Today, I am continuing these things discussion to complete the discussion about the protein synthesis. So today also our topic of discussion is protein synthesis. In my previous video, I was talking about the protein synthesis in ribosome. Actually, in prokaryotic cell, what happened? In eukaryotic cell, what happened? In eukaryotic cell, the mRNA will be produced in the nucleus, and this from this nucleus the mRNA will be transported into the cytoplasm of the cell, and in the cytoplasm there will be the ribosome, and in the ribosome the protein will be produced from this mRNA. But in case of prokaryotic cell, both the mRNA production and the protein synthesis will be happened in the cytoplasm itself, because there is no boundary around the nucleus. So there is no boundary of nucleus, so almost same thing, same location, the mRNA production synthesis and the protein synthesis happened at the same location, although it will be also happened in the ribosome, and rest of the things are almost same in prokaryotic cell and eukaryotic cell. So this has been I have talked about, and when I am talking about the mRNA from DNA template, the mRNA will be produced by a RNA polymerase enzyme. Actually, I have told you in my previous video like that there will be some initiating codon like AUG, GUG, and there will be some terminating codon like UAA, UAG, and UGA, Ochri, Amber, Umber, which is called Ochri, Amber, Umber. So, this initiation of the mRNA from 5' to 3' of the DNA template strand, it will be started by the RNA polymerase due to some factor, protein factor like sigma factor. This is called sigma factor. And when they will be terminated, the full gene will be terminated, then there will be some another factor, terminating factor will be attached to the RNA polymerase DNA complex, which is also called Rho factor. So initiation factor is the sigma factor, and the terminating factor, protein factor, is the Rho factor for mRNA production. And there will be no punctuation and pause in the full sequence of the gene, and one it will be started. So if there will be any variation, like this was the mRNA molecule, and this is the UGCCCUAA, like that. So here say that here this is started in the initiation codon, here is started the mRNA transcription, and it will be no pause, no comma, no full stop, and no punctuation is there. Or it will be read it from 5' to 3' direction in the in this way, so to complete the transcription of the gene. And if there is any it is started from, if it is not started from A, it is started from A U, then full thing will be altered. So your protein will be produced by this change in the sequence, starting sequence, it will be changed, and your protein will be non-functional. So in case of, I have told that in case of prokaryotic cell, in ribosome, this protein will be produced from the mRNA. This is the ribosome in prokaryotic cell it is 30S. Two subunit of ribosome 30S and 50S. And in case of eukaryotic cell, this will be 40S and 60S. 40S and 60S in case of eukaryotic cell. In case of prokaryotic cell, bacterial cell, it will be 30S and 60S. So actually this S, what is the S? S means Svedberg unit. S means Svedberg unit. Svedberg unit, it will be called in short form S. So what is Svedberg unit? Actually this ribosome particle will be precipitated. How fast it will be precipitated during ultracentrifugation? This unit is called the Svedberg unit. And togetherly, when it is in case of prokaryotic cell, it is 30S, and in 50S. Together, when we ultracentrifuge it, it will be 70S together. This will be 70S. So this is the Svedberg unit. Actually, in case of ribosome subunit, it will be discussed about that. And another thing is the I have told like that, and I have told that the three-dimensional when I am talking about the mRNA, let's complete me one thing, very interesting thing. In 2023, Hungarian scientist Katalin Kariko and American scientist Drew Wiseman together won the Nobel Prize in Physiology and Medical Science in 2023. Recently they have got the Nobel Prize for which work? For their unique work in invention of the discovery of invention of mRNA vaccine. It is very novel vaccine. First time it is used, this mRNA vaccine, first time for their research work, mRNA vaccine has been used for the treatment of for the vaccination of the COVID SARS CoV-2 virus, which is popularly known as coronavirus. Later, this actually this is approved by FDA, Food and Drug Administration USA, and this is the first time, and this is the only authorized approved mRNA vaccine. Generally, vaccine are produced from attenuated culture of the particular pathogens. Attenuated culture means their virulence will be not there, but they will be viable. Like that condition, this will be injected in our body to develop our immune system. So this is called attenuated culture. This is the traditional method of vaccination. But first time in SARS means severe acute respiratory syndrome, which is caused by the coronavirus. You know that whole world will be disturbed for this virus. We all know that, and this treatment for this corona pandemic will be controlled by this mRNA vaccine first time. And Moderna company has developed this virus due to the research work of Katalin Kariko and Drew Wiseman. So this is the very relevant factor. So I should discuss about this. So this is the thing, and I have told you how the protein has three structures: primary structure, secondary structure, tertiary structure, and quaternary structure. And how by these structures protein are actually folded. to its functionality, So if the protein fail to fold properly, then I have told you that two type of there will be maybe protein may be non-functional, or it may be toxic effect. It may be toxic. So like toxic, like prion, one I have told you, prion and amyloid. If the protein is not amyloid fibril, actually what is happened due to the prion is the one type of enzyme which will trigger the other proteins in the brain to unfolded, to not folded properly. Then there is the problem. And one of the famous disease for that deadly disease, mad cow disease, is due to the this prion, due to the not improper folding of protein. This and amyloid fibril, due to the, it is a soluble proteins, but it due to the soluble proteins assembled together to make insoluble fibrils, which will have some neuro disease effect like Parkinson's disease, Parkinson disease and Alzheimer's disease. Parkinson disease and Alzheimer. This is a very famous neuro disease. You know that Alzheimer disease. Due to this amyloid fibril, so protein should be folded properly to its tertiary and quaternary structure. Those protein have one polypeptide chain, they have only the tertiary structure, but those have the more than one polypeptide chain, I have told you, then the quaternary structure will be formed due to the hydrogen bonding and hydrogen sulfide bonding. Disulfide bonding. So this is the all about the protein synthesis. I hope that you like it very well. So thank you.